Hello and welcome to this video on the isoelectric point and gels. The last video on this channel was an oversimplified version of what the isoelectric point is and what it is used for. Here is a specific example of the isoelectric point and how it has been used with proofing PCR primers. As has been said, the isoelectric point of any given molecule is the sum of its parts, these having a range of charges, some being more or less polarized than others. This can be altered through a number of mechanisms. pH is the most common. An acid or base is added to the solution. This shifts the proportion of donor or recipient molecules. Another example used in biochemistry is SDS. This is otherwise known as sodium didecyl sulfate. It acts like a detergent in some cases and a buffer in others. In the case of proteins, it is a means of denaturing the peptide chain. This happens because SDS interferes with some of the intermolecular connections, specifically the non-covalent bonds. This changes the isoelectric point of the molecule. This in turn alters how the molecule operates internally, changing some of those other bonds because they no longer have an appropriate connection. One of the range of factors which affect this is the size of the molecule. Larger molecules generally have a positive charge, but they also have pockets of negativity which are hard to work around. This overall is known as their mass-to-charge ratio. Interfering with either the negative or positive aspect is one way of working around the charge of the molecule. This is an aspect that can be used for separating molecules from a larger solution. Case in point is the proofing of PCR primers. This combines the charge, size, and chemistry of the molecule to separate it along a gradient or against a barrier. Electrophoresis is an example of this combination of factors. The gel electrophoresis process begins by creating a matrix of agarose gel in a specially designed mold. Nominally, other chemicals are added to improve clarity and visibility of the gel bands. This setup is in a dock meant for electrophoresis. It has a bed for the gel to sit and two sets of electrodes. This attracts the relative parts of the molecules you are looking at. In the case of molecules with highly polar parts, these are drawn or slowed down more or less on the basis of their polarity. This gel block and apparatus are set up as a cradle with a bath around the gel block. A buffer is added to this bath. This will often be TAE, a shorthand way of referring to tris, acetate, and EDTA, in themselves a combination of chemicals. The product of PCR amplification is then added to the wells in the agarose gel with a DNA binding dye, along with a control sample. There is also something known as a ladder that is added to yet another well. This forms a quantifiable measure of the molecule size. This is useful if you want to know what size the product of your PCR amplification is, although in theory this should already be known. This allows you to identify them independently of knowing what well they've gone into. The gel is then charged with the electrodes that pull on the DNA. Since all DNA weighs the same, larger molecules would have to move slower in the agarose solution. The smaller molecules will naturally move faster as they are less impeded. If you have a lot of the amplified sequences that you are looking at, it will form a larger band regardless of how large the molecule itself is. If it has failed, you may not see a band at all, and if it is something that is expressed at a very, very low level, then you may see a relatively small band. It is all based on the predominance of that gene and how much it has been expressed. This is just an example of how the isoelectric point can be used in biochemistry to identify the presence and quantify protein, RNA, and DNA, as they are all subject to the same general principles. This kind of change can also play havoc with a wide range of other things that are more essential for life. For example, the cell wall and enzymes. This is exemplified by the earliest reports of gelphoresis, and these methods were being reported in the 1930s. Thank you for watching this video, and hopefully it has given you some understanding of how gel phoresis works and why the isoelectric point is important. Please post any comments, questions or suggestions you may have below.